Now you can go live, sir.
Yeah. YouTube live stream is start, Papa.
Okay, sir.
Now you can go live, sir. Yeah, sure.
Start a check. Start a
A very grand good morning to one and all, and welcome to the day 4 of FDP on nanotechnology in healthcare. On behalf of the Vishnu College of Pharmacy, I'm welcoming you all for this note, and I would like to introduce our director, sir, to the gathering, Dr. Kumar V.S. Nemani, director of HTCP since April 2018. He holds his B.Pharm and M.Pharm degree from Andhra University, Visakhapatnam. He has completed PhD degree under the guidance of ex-professor and director P. Ramarao Naipad Mohali. Subsequently, he was associated with Professor Jeffrey S. Mughil at McGill University, Montreal, Canada for his postdoctoral fellowship for duration of 3 years. Dr. Kumar V.S. Nemani has associated with leading research centers in India, namely Dr. Reddy's Research Foundation, Hyderabad, Piramal Life Sciences Limited, Mumbai, Lupin, Pune. He has over 14 years of drug discovery and development research expertise and managerial experience. Now I request our director, sir, to give the welcome address to the today's FDP. Over to director, sir.
Hello everyone. Welcome to today's national webinar on nanotechnology in healthcare. I'm pleased to inform all of you that the number of registrations are really good for this webinar series. The number is more than 1,000 registrations and the feedbacks are around 500 so far. This tells the importance of the impact of this role of nanotechnology in healthcare and understanding of the pharmacy community. I, on behalf of SVS Education Society, our chairman, sir, Sri K. Vishnu Raju Garu, our vice chairman, Sri Ravichandran Rajagopal Garu, and principal Dr. K. Prasad Garu, and staff. On behalf of all the staff at SCCP, I welcome each one of you for this webinar series. This webinar series is focused on role of nanotechnology in healthcare, and today's talk is more on COVID-19. And as all of you know, it is a pandemic and post-immune interactions plays a critical role, especially cytokine storm. Managing the cytostorm is a very, very important aspect of managing the patients affected with the COVID-19. I think today's speaker, Professor P.V. Brahmachari Garu is the ideal person to talk about on this topic. On this note, I thank all the principals and the senior staff who are supporting this webinar series. I'm sure that this series will be enriching and develop an interest, and you all get benefited with this. We also request each one of you to give our honest feedback. This will help us in improving ourselves and making this webinar series more successful. On this note, I also like to add one more point. Next week, we are conducting an FDP2 on drug discovery and development, and most of the speakers are from international expertise. I hope you do recommend it to your friends. So with this note, I thank Dr. Brahmachari Garu for accepting our invitation and being part of this webinar series. So once again, I welcome each one of you for this webinar. So thank you all. Over to Dr. Ashok.
Thank you, Director Sir, for your welcome note. Now it is my privilege to introduce the profile of today's guest speaker to the gathering. Dr. P. Veera Brahmachari Garu, did his BSc from Sri Krishnadevaraya University, MSc from Acharya Nagarjuna University, PhD from Goa University, DSc from Sambalpur University. And he had undergone a PDF at the Queensland Institute of Medical Research, Brisbane, Australia for a duration of 10 months. He done DVT PDF from IISc Bangalore for a duration of 2 years. Currently, he is working as the senior lecturer and head of the department of biotechnology at Krishna University, Machilipatnam. Added to his credit, he had published various papers and book chapters of after national and international reputation. And today he is going to deliver a guest talk on the topic host immune pathogen interactions and inflammatory response in the novel COVID-19 virus, with adding a note to the nanotechnology applications to the concept. Over to the guest speaker, Brahmachari. Sir.
Yes, yes. Yes, I'm sharing the screen. Yeah. Is my voice audible? All of you?
Yes, sir.
Yeah. So the most respected chairman of the Vishnu Group of Institutions and vice principal, uh, vice chairman, principal, coordinator, um, the teaching fraternity of the department and other people, organizing team. A very good morning to all of you, and I humbly thank all the organizing team to invite me for delivering a talk on this auspicious topic, which is very much needed of the hour. And thank you very much, Ashok Garu, for taking a lot of effort inviting me and organizing the seminar. Okay, so I think I have very less time, and probably I'll move ahead with the topic. And today I'm speaking on host immune pathogen interactions and inflammatory responses in SARS-CoV-2, which is a killer pandemic virus. And I also trying to link up because the theme of the seminar is completely nanotechnology, and definitely the vaccine preparations of COVID-19 will definitely involve a lot of nanomedicine preparations, especially in terms of protein-based nano delivery systems, because most of the epitope-based vaccines or DNA-based vaccines or RNAi-based vaccines are basically and maybe we'll need some nanoparticle delivery systems in order to deliver a vaccine into any human being who is infected with COVID-19. I hope my voice is audible to all of you. Hello? Hello?
Yes,
My
sir.
voice is audible?
It's very clear. Please go ahead.
Okay, fine. The first slide, this is the virus which I'm showing, which we are talking about the SARS-CoV-2, and we are talking about the vaccine strategies and nanotechnologies involved in preparing vaccines, and this is how lungs are infected. This is the basic target organ for the COVID-2 virus. Okay, so if you look into this slide, I must dedicate this talk especially to the people, to the laborers who have taken a lot of efforts in traveling to the workplaces, to their home places. Really, this was really a heartbreaking events which we have seen for the past few months in the televisions and the news channels. And definitely several people must have even died while getting back to their home places. It's very painful moment to the Indian scenario. And there's also one more slide which I'm showing so many pictures which were being depicted on the televisions, and very painful events. Okay, so let me come back to the history of pandemics. If you look into the pandemics, because if you look into the history, there are several viruses which really created havoc few million years ago. But still, what we are talking about is the novel coronavirus, which is located at the end. Okay, it's March 17, 2020. Okay, it's very small dot when compared to your HIV/AIDS and yellow fever and plague, cholera, smallpox, bubonic plague. Look at the size of the pandemics which took place. This is the history which I'm trying to show you.
okay, and definitely, and we may have to face much more kind of casualties, especially the deaths, the infectious disease, um, its control measures. There are so many things which we need to be aware of in the coming slides, which I would like to depict upon. So if you look into the transmission, the emergence, the evolution of these particular virus, that how it has taken place. It has taken place in different 5 stages. The stage 1, basically it is an animal virus. Uh, for say, we talk about the animal viruses generally infect animals only, and human viruses infect only humans. Okay, but now only after SARS-CoV uh, 2. Okay, so there is a big notion among the world that so there is a species barrier, species jumping from one species to another species, one genera to another genera. Okay, in stage 2, look at here, the animal disease reservoir where the virus naturally lives, bats, for example, for Ebola and SARS. Okay, so here there is a species jump from bats to other animals like civets, swines, avians, chimpanzees, monkeys. Okay, in the stage 3, we see the immediate intermediate hosts which are holding the viruses for a for incubation period for a short period of interval in civet cats. Okay, in stage 4, we see the transmission to humans and between the humans. The virus can adapt and mutate and can transmit between several host species. Okay, so what we are trying to say is, we are in stage 5 now. Now the virus has been transmitted. It's already in the community level and it's disseminated, and it's aided by increasingly mobile populations because there's a lot of mobile transmissions, human transmissions. Okay, a lot of transportations are occurring, and I'm pretty sure people are talking about 30 mutations which took place recently in India itself, and forget about and talk about the kind of mutations which took place in the Scandinavian countries and European countries and American countries, especially France, Italy, Spain, and other countries. Okay, and I'll talk about that in the later stage, especially how the database has been prepared and developed. Okay, and if you look into here, especially, so there are this coronavirus comes into coronaviridae. Okay, basically SARS and SARS-CoV-1 just come out in 2001 and 2, and SARS-CoV-2. We call them as both as cousin enemies. Okay, both are cousins. They belong to same clade, and basically these coronavirus comes under 4 types: alpha coronaviridae, beta coronaviridae, gamma coronaviridae, and delta coronaviridae. And the viridae which we are talking about basically comes under beta coronaviridae. Okay, if you look into here, the corona ancestors, basically bats and avians, and from there, there's intra-species transmission which took place here in the bats, the civets, small civets and avians, and here you can see interspecies transmission. From here, you can see the species jump from avians and to dogs, cats, swines. Okay, and from there, it has taken place to, it has transmitted to humans, and from humans, we see the lineages A, lineages B, C, and D. Okay, and in humans, we see the lineages of B, C, D. Okay, so if you look into here, this viruses basically come from the bats and pangolins. Bats don't get sick for 3 reasons. Basically, the evolutionary, if you talk about the evolutionary, it's not advantageous for the virus to kill large population of its hosts. The virus depends on them to survive and continue spreading. So what exactly happened is, for a period of a time, the bat has become attenuated for this virus and it's become asymptomatic, and it can harbor this virus but cannot be infectious, and it can transmit the virus, especially the COVID-2 from bats to other animals or other species. So the immune system fights the viral infections, but do not overreact to them, which protects the bats from the deadly hyperinflammation. So this hyperinflammation does not occur in the bats because it's already evolved along with the receptors, and it's already it already fought the immune system, and the hyperinflammation does not occur in bats at all. Okay, so if you look into here, SARS-CoV-1 has come up in 2002. The reservoir host is civet and cats, and MERS-CoV-2. All 3 are cousin brothers. Okay, MERS-CoV-2 has come up in 2012. It's a Middle Eastern respiratory syndrome virus. Actually belongs to coronavirus. Okay. And third one recently emerged is COVID-2. We call it as 19 because it has emerged in 19. Actually, COVID-1 and COVID-2 are 80% similar. Okay, only 20% they differ from each other. And we are talking about the humans and the chimpanzees. The humans and chimpanzees differ only with 0.5 to 0.8% genome in terms of the genome. But we are talking about 20% genome similarity. Okay, so 80% they are similar and 20% they are dissimilar. So look at the kind of mutations that might have taken place in the genome levels. And the reservoir for SARS-CoV-2 is pangolin, and reservoirs for MERS-CoV-12 is camels, and folies it is civets. And all now the COVID spill over to humans. Because there is all these three cousins of made sure that they can transmit the disease from different species to the humans. Okay, and here if you look at the casualties, the death cases are 774, and here death cases are 866. And this is the old data which I'm showing. The death cases have crossed more than lakhs. Okay, more than lakhs all put together. Even in India, the number is going pretty well. And it was in June 2nd, it was the cases in India reported only till something like 2 lakhs. And June 12th, it recorded like 3 lakhs 10,000. And now it is much more shooted up to 3 lakh 55,000 as on 2 days update. Okay, so if you look into the contacts and contact tracing, this is the major problem in India because we are thickly populated, densely populated Indian population, the second largest population after China. Okay, so we are not sure about because Indian population, we are having a lot of cultural, traditional, and so many outlooks on all of us. But exactly what happens is Indians are very poor in disclosing the information to the healthcare and the medical systems. Okay, to the public health perspectives, where took enough efforts to have deployed revenue, police, medical, healthcare staff in nook and corner of every houses, but the Indian population has not reported, has not informed about their primary and secondary stage of their infection. They have contacted the hospitals only in the last stage of the infection. That is how several deaths have occurred, especially in India. And even now, people does not want to disclose any personal information because they feel that they become stigmatized among the society or among the public, and they don't want to inform anybody. So, and they want to keep it within themselves. Even they're ready to die, but they don't want to disclose even to the neighborhood. So this is how the imported cases came from abroad to India. And these are primary contacts. Okay, it's a local transmission, and secondary contact is how the local transmission took place to several societal, like churches, temples, and social gatherings. Okay, from there now the community transmission has taken place, a tertiary contact, and now even it is even crossed with quaternary contact system. Okay, so if you look into here, what is contact tracing? It's an alternative to prevent long-term lockdowns or overwhelmed health systems. Okay, if you look into here, what Indian country, the whole world is looking is, so we need to figure out the people, the COVID cares, okay, in the not infected stage, in the very whenever you have the preliminary symptoms like coughing, sneezing, fever, okay, immediately you have to contact the healthcare system and report to them, and you can put them in the isolations and quarantines, and you can put them in very contained centers. Okay, so this is there are 3 levels mentioned here. If you can pick up at this level, okay, if you do not pick up at this level, the disease is going to transmit, okay, from the patient 0 to patient, so many patients. And the second level, okay, at least we can limit within one particular thing, okay, we are one step behind the virus. And here, this is what exactly the government is looking out for. The transmission should not occur and spread to the public, which was not anticipated, and now it has already spread to the public. People are thinking that in July or August, probably it may even cross 3 or 4 lakhs, or 4 crores, or 5 crores, or 3 crores. That's what the people, that's what the healthcare professionals are estimating, because still the people are not aware, and they're almost all every day they are on the roads, and and they are not aware about this thing. Because I am telling all this because I worked in the virology laboratory in Indian Institute of Sciences in Australia. Because I preliminary worked completely on the virus, and I worked on the Streptococcus bacteria and this virus specifically, because that is how I came to know much information about these viruses. And I'm very much interested to disseminate the knowledge of these coronaviruses. So if you look into contact tracing and community monitoring, these are the major drawbacks in Indian scenario. So healthcare professionals, even they go to homes and they are unable to pick up individuals from urban environments, and there are so many unknown contacts. Okay, there are symptoms similar to other diseases, hospitalization. There's so many things like susceptibles and exposed people, infectious and remote people. Lot of confusion even till today. Okay, so the communities with infected individuals monitored daily, but effective due to early identification of infections is there. People are not disclosing any information. So this is where we are. The Indian scenario, as Indian population is really extremely going back, and popping up several infections day by day. So this is how the virus spreads from patient zero. Patient zero infects two people, and they infect two people, and two people infect four people infect 16, 16 will infect, like just like that. It goes on a logarithmic um, okay, it goes in a logarithmic shooting up of okay. The average of the people that person with the virus infected is on the R naught scale. So in India, people are not disclosing about R naught scale. Because if we talk about the R naught scale for SARS virus, it's 2 to 4. And if you look at the R naught scale for COVID-19, it's 2 to 2.5. MERS, it's 2.5 to 7.2. H1N1 is also one of the biggest pandemic, and it is 1.2 to 1.6. Only a small number of people got infected. And now, probably this R naught might have already increased to 3.5 to 4. This is what is my guess. Okay. So if you look into the some of the ethical considerations in India, especially contact tracing is a public good and has been used to reduce the risk in this century. And this tracing technique must strike a balance between the individual rights and the moral rights. And these tracing technique need to be very sensitive to this balance and attain the privacy and confidentiality when interviewing cases and contact tracing. And technology tools can assist making them by creating Arogya Setu apps, and several apps are also coming set up by the government of India. Okay, so many traditional contact tracing efforts are put it, and so many volunteers are deployed in order to find out the cases. So if you talk about the death in United States, so the most two common types of medical errors that took place, especially there are so many myths and misconceptions occurred due to usage of hydroxychloroquine. And now yesterday, some people have come out with the dexamethasone. That particular treatment is pretty good. So if you look into the kind of medical errors, okay, after heart disease and cancers, especially the medical errors stands third place in the world. These kind of medical errors, in the sense, hospital acquired. You go with one infection and come out with the 3, 4 infections. Okay, hospital acquired nosocomial infections, missed or delayed diagnosis. Indian scenario, okay, Indian hospital system is especially lacking in molecular virology, molecular diagnostic systems. No hospital is having a quarantine or isolation or a virology molecular diagnostics laboratories. There are no experts in India, especially who are expertised in doing real-time PCRs and quantitative qPCRs, other PCRs, because these viruses cannot be detected with the kids. And whatever information we are getting with the kids is just with immunoglobulin M and immunoglobulin G, which is highly unpredictable, and we get so many errors in that. And avoidable delay in the treatment. This avoidable delay in treatment we have seen in Bombay hospitals, and even till today they don't have hospital beds. In an inadequate follow-up and after treatment, inadequate monitoring of the procedure, and failure to act on these results, especially the diagnostics, is a major drawback in the Indian system. Okay, so existing medical conditions and risk factors. Look at here, people, especially who are already suffering from diabetes, hypertension, and COPD, asthma, emphysema, chronic obstructive pulmonary disorder, heart disease, liver disease, kidney disease, weakened immune system, immunocompromised people. including a weakened immune system, or a steroid or other medications that affect the immune system. Okay, so these people are highly susceptible. If these people will be firstly infected if they go outside. So people, especially with any of these ailments, if they go outside, okay, they should be very cautious because their mucous system, mucous layers will be less compared to other individuals. So I'm talking about the genomic epidemiology of this, especially the virus, especially what exactly happens is this database is maintained by Next Strain team in UK. It's maintained by WHO. So what exactly is happening is, so today we have got so many cases. We have got, for example, 100 cases. Okay. Within this week itself, within a week days, okay, that particular genome has to be sequenced, and that genome has to be submitted in the central database in in USA, wherever the WHO is located. Okay, if we do not submit the genomic sequence database, we do not know at which place, at which particular place, this there is a mutation occurred. Okay, there is a recombination occurred between the genomes or between the strains or between the serotypes, between the strains, between the variants. Okay, so and other countries, especially China and other countries, even underdeveloped countries, are also doing pretty good job, especially in submitting the sequences to the central database. And I'm pretty sure we have got only one institute in India, that's National Institute of Virology, where that institute is unable to take up the load, the whole country, to sequence the samples and at least trace out where exactly is this mutation occurring. Is it occurring only in the receptor or on the spike region, spike protein 1 or 2, or ORF, or which antigen? Is it occurring on the lipid bilayer? We don't know exactly. Okay, but people want to know because only if you compare the central database systems, especially in the whole world database, then only we can come to know where exactly this particular virus is getting mutated. So which population is being susceptible to these viruses? So if so, is it moved to S1 region or S2 receptor or somewhere else? Okay. So this is how we can come to know if you submit the whole gene sequence database to the central database, we can come to know how the first initial strain from Wuhan had traveled from Wuhan to other countries, how the blue line is traveling, how the red line is traveling, how it's moved from United States, how it's moved from Spain or France or Portugal or other countries, especially Paraguay and Ecuador or all kind of Scandinavian countries. If you look into here, there are 4 clades, especially where the disease transmission has taken place. Okay, and this pocket of Scandinavian countries, because these conditions are very cold there. It's the northern poles, the Scandinavian countries, because here this particular hamlet of this region is also infected pretty much, and especially this region of America is also infected pretty much, and it has even gone to Congo, Nigeria, Brazil, Peru, Bolivia, Paraguay, Argentina, okay, even gone to Australia. All this clade of Australia, Papua New Guinea, Indonesia has moved from China, even to India. Okay, some part of people have moved from China to India. Okay, so it's like that. So if you look into the kind of, I was talking about the genome sequences. If you look into here, the SARS-CoV-2 and pangolin and bats, if you look into here, this particular region is a polybasic cleavage site, O-linked glycan residue. This is the place where it comes and binds to the ortho-linked glycan residues in our mucous layer. Okay, so this is the place it's mostly mutating. This place, PRRA, this particular region of this 681 to 684 region is highly mutating. And if you look into here, there are 2 kinds of circulating SARS-CoV-2 strains across the world. Okay, there are L type and S type. L being the major type, this is come from China, and then that particular clade has moved to US. So we don't come under L type or 70%. We come under only under S type or 30%. This is a minor type. We come under these. If you look into this part of this region, these are so many isolates. Okay. If you look into here, L, look at here, L, yes. Okay. And this particular region is being highly mutated. So one single point mutation, one amino acid change, there's a lot of difference. Only one amino acid change can become a new serotype or a new variant, and absolutely, okay. So that may even recombine with the other circulating strains in the world and environmental strains, and then recombine and become a new serotype. And if at all, what happens is that particular mutated strain, if it jumps the species barrier from humans to another species of animal which has not infected before, so again, in that particular animal, it's going to mutate again, and it may even cause much more devastating effects, because we have seen only few couple of lakhs of casualties and deaths. But if at this stage, if it jumps the species into new species from humans to other species now, and comes back to humans again, with so many modifications in its genome. Okay, I'm pretty sure we may not be able to handle this strain or COVID-2, and again one more new strain which is coming, which may come up in the coming years. Okay, so if you look into the this was just a clip picked up from the BBC News. I took it from my mobile phone. Look at here at 3:12. Okay, and 3:30, a small, just 1 minute, out of 1 minute, you can see the number of deaths. Okay, so death is just like your environmental status. Okay, the death toll is being increasing every minute across the world. Okay, so look at here, deaths 2247, and here if you look at the deaths, okay, 1813 only in China. Elsewhere you see so many other kind of deaths. So every minute, because when we see these outbreaks of pandemics, okay, this really changes the scenario of the death cases, the casualties. Okay, so every minute and every second it changes. Okay. So let me compare the genomes of COVID-1, and which has come up in 2002, and COVID-19. The spill reservoir is known. We know the spill reservoir pretty well in COVID-2, in COVID-1, but spill reservoir is still unknown. We are unable to determine whether it's come from bats or fungi. We don't know, because we don't have enough database of pangolins or bats or human viruses. Okay, mode of transmission, most transmission occurred in hospital, but here widespread community transmission is there. There's a lot of difference between these, and no transmission until 24 to 36 hours of symptoms, no asymptomatic cases. And here, so many people in India are asymptomatic. Possible asymptomatic transmission is there. So many people in India are harboring the viruses and unable to show up any symptoms, and they are called as asymptomatic, and they are most dangerous than anybody else. So, and the greater affinity of SARS-CoV-2 for the host receptor, okay, and increase in global travels, the global traveling, the economy, the system is not come down, and people are worried about the business, the economy. Okay, people are not worried about their health consciousness at all. Okay, so they do not know whether they are transmitting the virus, they are acquiring the disease, and they're in turn spreading the disease in the community level, and they're also losing their family members. So if you look at the structure of them, probably all of you might be knowing, it's like a crown-like structure, and okay, that is how it's called coronavirus. And there are so many membrane proteins, envelope proteins, spike proteins. So this is the actual culprit, spike protein, which I'll mention at a later stage. The diameter is like this. Okay, the two viruses, the ribonucleic protein is inside, it's a lipid bilayer. Okay, and the key concepts, there are so many virions and capsid proteins, and okay, so it's like crown-like appearance. The genetic material is RNA, not DNA. It is negative standard RNA viruses. Okay, and if you look at the structure of viral proteins, look at here. There's so many structural proteins in domain 1, domain 2, domain 3. There's so many structural proteins. Even a small change in one nucleotide to one amino acid, okay, the entire host pathogen interactions, okay, entire complexity will be changing, entire immunity dynamics will be changing, immune activation will be changing. Okay, even your vaccine, whatsoever vaccine has come up, cannot go and bind, even if there is single amino acid change or a single nucleotide change in any of the genome. So people are scared about the mutation which the virus is going to acquire in the days to come. Okay, so if you look at the genome, it's just 29 to 30 kilobase pairs. It's a very small genome. Okay, there are only 2 open reading frames, and when I read into the literature, there are only 29 genes in the SARS genome. Out of 29 genes, and we have got 30,000 genes, and 30,000 genes for 30,000 different proteins. And what exactly is happening is in viruses, in COVID-2, out of 29 genes, 27 genes of virus is interacting with all 30,000 genes in the human beings. This is the major problem. And this kind of scenario has never occurred in any other viruses. This is the first time these 27 out of 29 proteins are interacting with every protein in the human beings, and we do not know what exactly is happening. Whatever we knew till today is only hyperinflammation and hypoxia and a cytokine storm. Only these three things people could find out till now, but we do not know what else this virus is doing inside the human cell. Okay, so then if you look into here, uh, S protein is divided into two types, S1 and S2. S2 determines the virus host range and cellular tropism. Okay, so S1 is a very peculiar protein and the spike protein, and S2 mediates the virus cell host fusion. Okay, by HR1 and HR2. So these two are very important in attaching to the ACE2 receptors. Okay, and host, there are so many host factors, host pathogen interaction factors. Okay, human angiotensin converting enzyme ACE2 receptor. I'll talk about that. Okay, so mostly people who are susceptible are people about 65 years of age and people with underlying diseases or immunocompromised patients. And cytopathic effects, cytokine storm and sustained inflammatory responses, hypoxia, septic shock, etc. may be related to critical conditions. If you look into here, hypertension, COPD, diabetes, cardiovascular disease, respiratory distress syndrome, septic shock, metabolic acidosis, coagulation dysfunction, multiple organ failure, all can be noticed in COVID-2. Very complex viruses which we must not have ever seen. Okay, and look at here the kind of binding. All the bat, okay, even the bats are going and binding the ACE2 receptor, swine's ACE2, civet ACE2, mouse ACE2, everybody are competing for the ACE2 receptor. I'll talk about the ACE2 receptor in the coming slides. Okay, so there are nearly 16 non-structural proteins in NSP1 to NSP16. Okay, the positive stranded single stranded RNA. Okay, so there are only two ORFs, ORF1A and 1B, and there are 29 genes together, and only 27 are functional and 2 are non-functional. Okay, so is my voice audible? Hello? Hello? Is my voice audible, sir?
Yes, sir.
It's very clear. It's going good. Please go ahead.
Very good. Okay, fine. So when we talk about the human mucous membrane, see, in our human system, in the saliva or in the epiglottis region, we have got mucous layer. The mucous layer is just like a silky kind of thing. Look at the funniest part. The God has given us a very wonderful system of mucous layer. It is 3-layer: mucosa, submucosa, and adventitia. Okay, it's not that easy. The picture which I'm showing it here is nothing but this particular region. Okay, there's so many cilia. Okay, so it has to cross several barriers: lamina proposia, bronchial gland, smooth muscle, cartilage, connective tissue. So there are so many layers. Virus just can't get inside, because I'll talk about that. What exactly happens? These are globate cells. Okay, basement membrane. Look at this is how the virus gets inside. Okay, it's a tonsillar lymphoid tissues, tongue and palates and cervical lymph nodes will attack initially. Then Dendritic cells, B cells, and everything, macrophages will come into picture here. Okay, esophagus, trachea, bronchia, okay, and bronchial lymph nodes. This is the ultimate target region. Alveolar macrophages, the bronchial lymph nodes. Okay, so what exactly happens is this. Remember this cilia. Okay, so virus-assisted bacterial invasion in the lung. Just look at this slide A. Okay. So this is the mucus which I was talking about. This mucus which I'm talking about is a mucus layer. So now at A, virus acquisition takes place. Okay, and this virus is picked up by the this orthocealic acid linkage located on the mucus layer. SA cealic acids. Okay, this cealic acid is very peculiar. Okay, so only neuraminidase enzyme on the virus, the spike proteins, can cleave the enzyme. It's called as molecular scissors. Okay. So only these can cleave it, okay, and it can cleave it, okay. Only after cleaving, there are so many bacterial, okay, relatives, neighborhoods of viruses. They are ready to enter, but no bacteria till now has got a mechanism to open or cleave the cealic acid linkage. Only H1N1, flu virus, and coronavirus have the capability, and no other viruses got the capability to cleave the cealic acid linkages. Okay, so I'll tell you how it cleaves. And this is where the tissue is located. So it has to enter inside the tissue, and tissue invasion occurs. Okay, and look at here. Once, hello.
Sir, it is audible, sir.
So once if there is once if there is, once if the viruses cleaves the neuramic acid, there's so many bacteria enters along with the viruses and then gets inside the tissue. And here there is something called fibronectin binding proteins. Okay, so bacteria has got a capability to cleave the fibronectin binding proteins, bind to them and cleave them and easily get in it. Okay, but bacteria has not a mechanism, has not got a mechanism to cleave the neuramic acid. Okay, so if you look into here, so I think most of you must not be aware this virus, this spike protein, and this is a cilia which I'm talking, and this virus particle is spotted like a pink spot on the cilia. Okay, see, look at our cell. Okay, as far as my knowledge goes. Okay, plant carbohydrates, animal carbohydrates, mammalian carbohydrates, bacterial carbohydrates, yeast carbohydrates. You take any carbohydrates across the globe, all the carbohydrates are synthesized in the cytoplasm by glycolysis. My knowledge goes. Okay. But here in the human system, wherever the mucus is there, in all the eyes, nose, nasal fluids, mouth, and all the openings, okay, wherever the mucus is there, okay, this mucus in the human system, mucus is nothing but orthosialic acid, it's a long chain polymeric carbohydrate substance. Okay, this particular carbohydrates are synthesized inside the nucleus. Okay, so that is the reason. This is I'll say this is a God's gift to human beings. This is the reason why no other bacteria, no other viruses except corona and H1N1 can cleave this sialic acid. Sialic acid linkage is not that easy to cleave. It's not that no chemical or no enzyme till now has cleaved, has no reported cleaved sialic acid. Okay, so it can be prepared with so many post-translational modifications and post-transcriptional modifications, and so because it's a glycoprotein and it gets deployed into the nucleus, and from there so many ubiquitinalizations and so many things occurring. Check post stamping inside the nucleus. It comes with a secretive code like molecules, decoy molecules, and gets inside the cilia and gets inside the cytoplasm, gets outside the cytoplasm. Okay, so this is how it can see. Look at here. So this is cytoplasm. Okay, and this is so many carbohydrates. Okay, and these carbohydrates are getting inside the nucleus and making all the silyl transferases and getting so many modifications inside the nucleus. And these modifications are very much secretive and nobody knows, only the genomic DNA, only the DNA knows what kind of modifications it's making inside the nucleus and comes outside the nuclear pore with the help of so many silyl transferases which can transfer uh, the modified carbohydrates from the nucleus and then gives to cis Golgi, trans Golgi, and from there initially to the endoplasmic reticulum and then cis Golgi, trans Golgi, and with the help of vesicular endothelial transports, okay, clathrin mediated endotransport endosomes, it goes and transports to the orthosialic acids or in the form of mucus or in the form of cilia. Okay, so it will go and transfer into the extracellular space from the intracellular space. Okay, so if you look into the barged entry, I'm talking about people are talking only about the virus and virus and the virus. But as far as my knowledge goes, virus, if your immune system is pretty good, virus gets inside, it does a job, okay? After 14 days, it gets outside. It does enough of damage to the system. And after 14 days, it will get outside the body. But bacteria is the actual culprit in terms of SARS-CoV-2 and COVID-1. What exactly is doing is bacteria, so many bacterial pathogens are most deadly compared to viruses. Okay, bacteria will not start its pathogenesis until 14 days. Okay, um, it will stay inside the liver or the lungs for a period of time, and only before the entry of the viruses, before the exit of the viruses, the bacteria, especially Staph aureus, Streptococcus pneumonia, okay, then Streptococcus pyogenes, Candida albicans, and so many people who are very much anxious, like especially mouth pharynx respiratory system is colonized by these people, and skin is colonized with these bacteria, and stomach is colonized by these bacteria. So there are so many bacteria coming along with the virus after the sialic acid breakage. Okay, So they gets inside and then they start the whole mechanism that takes the control of the whole host cellular immune responses, and then they'll start mimicking and hijacking the whole signal transduction system in the human body. Okay, so let me talk about the invasive bacterial virulence factors. There are so many bacteria: Klebsiella pneumonia, okay, then Streptococcus pneumonia, Staph aureus, E. coli, okay, E5201 strain, then Streptococcus strain. All these can evade the host immune defenses very easily because right from the beginning, virus has took along the bacteria inside the system. So this is the CDA which I was talking about. Okay, the host invasion strategy. This is the bacteria which I'm talking about. This is a bacteria. These are also even bacteria. Okay, so in this slide, exactly what happens is, along with the virus, this bacteria comes inside with a very few numbers, one or two numbers. So initially they make themselves as pioneers, then settlers, then they become a society. Okay, they start sensing among themselves with the help of quorum sensing or exopolysaccharide production or bio surfactant productions or bio emulsifier productions. Okay, they start preparing some slippery molecules around themselves, the mucus molecules, and then they get to learn, they get to start studying the cell signaling, coaggression, metabolic synergy, genetic exchanges, and then the society becomes a community. And once they become a community, no antibiotic, no doctor, no bacteriologist, no virologist can save anybody from that. So we are just talking about the viral outbreaks in the coming years, I'm pretty sure. Okay. Already there are so many infections in the hospitals with nosocomial infections, okay, which there is no treatment, and the bacteria are even become multi-drug resistance has already gone long back, and now it is extremely drug resistance which has been taking place. Okay. So this is the next um, mm, big uninvited situation which Indian country is going to face. So when I show this slide to you, let me go back and show you this one here. This is slide A. Okay, look at here. The virus acquired, and it has not gone into the tissue yet. It has just crossed the sialic acid, and look at here in this level, in this stage. Okay, and now it has crossed the tissue. The virus got into the tissue, and macrophage impaired functionality. There's so many dendritic cells are coming there. Okay, and interferons are coming here. Viral infected tissue. Now it's become a viral infected tissue and bacterial infected tissue. So both, along with the virus, even bacteria is also there, but nobody's talking about this. Okay, interferon alpha, interferon beta. Okay, there's so many natural killer cells, recruitment, and so many decoy molecules are deputed or deputed by the immune system to the site of action. Then so many neutrophils, PMNLs, polymorphonuclear lymphocytes, leukocytes is deputed there. Then viral infected neutrophils, and there's so many cells are getting apoptosis. Programmed cell death is taking place. So many healthy cells are dying, and it's almost crossed the endothelial layer. And now, once it enters into the bloodstream, okay, after tissue, bloodstream, okay, if it enters the bloodstream, it becomes viremia, and a person may die. And if the bacteria enters the bloodstream, it becomes bacteremia or septicemia. Okay, and people may die. And people whomsoever have survived today after infection, okay, those people are not nobody, no other than this virus has not crossed the blood, has just stayed in the tissue, and these immune cells have revoked and fought against it and thrown them out of the system. Okay, so in India especially, we have got a lot of melanin content in our system, and we are basically we have got very good anti-defense systems, and people say that all the Indians have got huge amounts of HNRNA. Okay, HNRNA in our cells, and these HNRNA is protecting against so many viruses and bacteria. So that is also one of the greatest gift from the God to all the Indians, especially whomsoever are black like me, dark like me. Okay, so if you look into here, how coronavirus hijacks your cells, how it's going to hijack or kidnap the signal transductions, hijack your pathways. Okay, so I'll talk about this. So virus is covered with the spikes and then enters into the vulnerable cell with the help of ACE2 receptors, and then releases the viral RNA and hijacking the cells. Then after enters inside, it hijacks the host cellular protein machinery and genomic machinery, and they start preparing their own viral proteins and viral RNA molecules, ribonucleoproteins, and they start making their own assembly of new copies, and then they spread the infection. Then immune responses completely collapsed. They leave the body, okay, and when they leave the body, they'll snatch away some amount of host cellular membrane, wrap them across the cell, and gets outside, exits out of the cell, and then mimic like a human cell whenever it infects the next cell. And the human cell is unable to demarcate or distinguish whether it is a self-owned cell or a foreign cell. Okay, because it is already having a human cell membrane, lipid cell bilayer membrane, the human cell will very well accept it as a non-self antigen or a non-self entity. So if you look into the stages of COVID-19 infection, stage 1 is asymptomatic, and we are asked to only wash your hands with a sanitizer, but we are not asked, we are not told to gargle at least 5 times a day with turmeric water or with a salt water, because that creates some kind of a osmotic pressure on the virus cell, and will not allow the virus to go and attach to the epiglottis, the small tongue, and the mucus layer. Okay, and this has to be also maintained, especially in all the Indian individuals. And in stage 1, inhaled virus likely binds to epithelial cells, the nasal cavity, and starts replicating. And direct membrane fusion between the virus and the plasma membrane of the cell. Angiotensin converting enzyme ACE2 is the main receptor for both SARS-CoV-2 and CoV-1, and local propagation of virus, but a limited innate immune response. We have very limited innate response because we don't have enough memory cells like toll-like receptors to fight against CoV-2, and viral burden may be low, but however individuals are very infectious, and for that particular period of 14 to 21 days, that particular human being will undergo so much of oxidative stress and so much of cellular stress. So then nasal swabs might be more sensitive than throat swabs. That's the reason why people are taking nasal swab, because nasal swab has got a lot of mucus and it can entrap the virus while inhaling through nostrils. And stage 2 is in stage 1, it can stay up to 1 or 3 days in epiglottis, and stage 2, upper airway, connecting airway response. The virus propagates very fastly and migrates down the respiratory tract along the airways, and a more robust innate immune response is triggered, and the nasal swabs and sputum may yield the virus as well as early markers of the innate immune responses. And COVID-19 disease is clinically manifested. The level of cytokines are the same as other innate response cytokines, may be predictive of the subsequent clinical course for more aggressive monitoring. And for exactly more than 80% of infected patients, the disease is mild and mostly restricted to the upper and conducting airways, upper respiratory tract and conducting airways. And because for other viruses, if we talk about H1N1 and flu, another virus, it's only causing infection only in the upper respiratory tract. But COVID-2 especially is infecting both upper respiratory tract as well as the lower respiratory tract. There is a major problem, major drawback with the COVID-2 virus. And stage 3, look at here. So all the oxygen is depleted in the cell, and the cell is deputing all the cells into the site of action, and there is no enough oxygen in other cells, and also oxygen is depleting. The condition is known as hypoxia. So infiltrates and progression to ARDS, acute respiratory distress syndrome. Only 20% of the infected patients may enter into stage 3 and develop pulmonary infiltrates, and some of these will develop very selective disease. The virus now reaches the gas exchange units of the lungs, infects alveolar type 2 cells. There are two kinds of cells, type 1 and type 2. Okay, so it goes and infects only type 2 cells, and SARS-CoV-2 propagates within these cells, and large number of viral particles are released, and the host cells undergo apoptotic cell death. Okay, the aberrant wound healing may lead to more severe scarring and fibrosis than other forms of ARDS. Okay, so recovery will require a strong innate and acquired immune responses with epithelial regeneration. So most of the time, people who are badly infected does not have epithelial or endothelial layer regeneration. So that is the reason why enough surfactant is not produced in the epithelial lining of the lungs, and because if there is enough surfactant lubrication, okay, it can be pumped outside, but there is a tissue damage in the lungs also. That is the main reason. So this is how there is a alveolar type 2 cells are getting infected. And see, look at here, that these cells are getting broken after the viral infection and release in the host cells under apoptotic cells, especially the lung endothelial cell lines. Okay, and if you look into this slide especially, so this is the receptor. Okay, so host DNA and its viral RNA, okay, gets inside pyroptosis, not apoptosis. Pyroptosis means body will have severe temperature and okay, cells will undergo pyrosis, okay, pyroptosis. Then viral maturation takes place here, and then next stage is it secretes so many interleukin cytokines. This is a positive feedback. If it is a positive feedback, what exactly happens is, okay, leakage is caused by vascular permeability and so many, okay, dysfunctional immune response. If there's a dysfunctional immune response, okay, so so many cells are secreted, okay, and the systemic cytokine storm, pulmonary edema, and pneumonia is formed. Widespread inflammation and multi-organ damage is also formed here. Okay, if there is a dysfunctional immune response, if there is a functional immune response, if there is a healthy immune response, infected cells rapidly cleared, then virus inactivated by neutralizing antibodies. So we don't have enough neutralizing antibodies against COVID-2 because we don't have enough memory cells for these things. Okay, so unless and until if your immune system is pretty stronger compared to other people, because people who are smokers, people who are alcoholics, and people who are already having immunosuppressive diseases or immunocompromised, they're already taking immunocompromised diseases. If they have underwent any surgeries, okay, they must be under high threat, and their immune responses will not work. And here, for deploying, mostly alveolar macrophages will recognize the and recognize them, phagocytize them, and depute the apoptotic cells and kill them with the help of neutralizing antibodies, and then activates the virus and exits them from the system. Okay, so immunological imbalance. Okay, so significant reductions in CD4, CD8 cells in the blood of infected patients. Okay, this recently reported in Nature Medicine, and report in Lancet shows ARDS is a common immunopathological death event. Is reported in Lancet 2020, and main mechanisms for ARDS is the cytokine storm. Cytokine storm. I'll talk about this. A deadly, uncontrolled systemic inflammatory response resulting from the release of large amounts of pro-inflammatory cytokines. Inflammatory cytokines are very good, but more is not good. Deputing from other cells, borrowing the cytokines from other cellular organs, tissues is not good because they will be deployed, they will be deprived of this system in their cells, and especially cytokines, interferon alpha, gamma, interleukin 1, interleukin 6, 12, 18, 33, tumor necrosis factor alpha, TGF beta, okay, and chemokines, CCL2, 3, 5, 8, 9, 10, and all these are immune effector cells. All these are deputing to cytofaction, and there, just like your thunderstorm or tufan, okay, your heart, your lungs is full of cytokines. Your lungs is completely invisible in any of the X-rays. These cytokine storm can trigger a violent attack by the immune system of the body, causes ARDS and multiple organ failure, and finally lead to death in the severe cases of SARS-CoV-2 infection. So these findings may provide valuable information for the rational design of vaccines against SARS-CoV-2. So we talk about how the lungs are getting affected. Look at here. This is the healthy lungs. We have got red blood cells in the capillaries. Okay, we've got type 1 and type 2 alveolar cells. And look at here, the layering, the surfactant layering is there in the alveolus. Okay, and there's a lot of exchanging taking place between oxygen and CO2, inhaling, exhaling. Okay, here is a major problem. Surfactant and endothelial layer. This virus is killing away all this surfactant layer, endothelial layer, and which is not regenerable. Okay, generally it has a regeneration capacity, but virus is attacking so badly that the surfactant and the endothelial layer cannot be regenerated in the epithelial endothelial layers. Okay, this is early stage. The virus enters inside the immune cells and alveolar sacs. Okay, so here so many cytokines are deployed. Too many immune cells create a hyperinflammation in the air sacs, and layer of dead cells makes breathing impossible, and that is why the people are dying on the ventilators. And what I say is, this is my notion, because most of the ventilators in India or abroad cannot be sterilized, and so many pathogens these ventilators will make it a home for so many bacterial pathogens, because if you cough, if you sneeze, will go and stay inside, and they do not have very good ample aseptic system to clear them. So for that reason, if you go with a viral infection, you may acquire bacterial infection through ventilators. That's one of the major drawbacks. Okay, so this is how the protein-rich fluid is there inside. All this protein-rich fluid is uh, getting depleted. Okay, all this cellular debris and all this surfactant is lost, and fluid is filled with interstitium and neutrophils. Okay, and there is no enough space for exchange of gases inside the lungs. Okay, so the SARS-CoV-2 when infects with the help of ACE2 receptors, genomic release. Okay, then host cell ribosomes and proteolysis, then replication, translation, multiple proteins, and they're released outside, and then they go and start infecting. So there are so many things like, so once they infect immune classical, complex classical pathways, there's so many cytokines, the complement system is activating, interleukins, thermonecrosis factor, CCL2, chemokines, cytokines, neutrophils, macrophages. All this will accumulate in one place. They are called as cytokine release syndrome and accumulation of neutrophils and monocytes and macrophages. They are accumulating, but they cannot function because virus killing is a different kind of is employing different kind of a strategy. So look at here in day 1, virus is entering ACE2 receptors, okay, and day 2, day 3, even if you give remdesivir, ritonavir, lopinavir, nothing is going to work. And here complement is getting activated. Okay, so I think activation of innate immune response in the lung will start only in the 5th day, and 5 to 7 days. Okay, we can give it anti plasma therapy in the form of IL-6 or IL-7. Okay, if it crosses 7th day to 14th day, a person is going to ARDS, acute respiratory syndrome, and then likely to enter into bloodstream and die with a viremia, whole body viruses. Okay, so the host niches, especially targeted by invasive pathogens, but here the gastrointestinal tract also. Okay, so from lungs, even if you go into gastrointestinal, because even gastrointestinal tract has got mucus lining layer. Okay, and you go inside, even if you can go inside the skin, you can go inside the oral cavity, teeth. Okay, you can go inside the respiratory tract, you can go inside the urogenital tract, vaginal tract. Okay, so wherever there is mucus, this virus has got the niches to target an invasive pathogens. Okay. So these invasive infections spread and target other tissues, especially blood circulatory system, sepsis, and viremia. Okay, people say that it has already crossed a blood-brain barrier or tissue-blood barrier. Okay, blood-brain barrier and olfactory portal, and people are saying that it has got a peculiar habit of, if you're infected with the virus COVID-2, peculiar habit of losing the sense of smell and sense of taste. As completely loses at least for 3 months. You cannot sense because we have got the G protein coupled receptors, nearly like 650 receptors, only to sense different smells, taste, and sensory organs. Sensory receptors are there in our body. So all those receptors are getting affected with the help of viral infection, COVID-2. This doesn't happen with any other viruses. Okay, so COVID-19 and blood clots. Look at here, so many blood clots are occurring. Okay, blocks the blood flow to extremities, and clots. Look at these kind of systems. The thrombotic events are also taking place. You see so many Dutch patients. Okay, several people died with blood clots after infections. Okay, so frontline medics reporting there are so many complications with reference to your blood clots. Okay, so the major drawback in coronavirus. So I told you there is a cytokine storm and ACE2 suppression, ACE2 receptor. Okay, the suppression of these causes both direct and indirect myocardial injuries. Okay, microangiography, diabetes, myocardial infarctions is going to eat away all the troponins in the patients with the known CADs, and inflammations and viral infection cause plaque instability. There's so many things are occurring. Okay, with immune-mediated injuries or oxidative stress, endothelial dysfunctions, and altered perfusion, vasospasm, and endothelial dysfunction of plaque instability. There's so many things happening with the stress occurred due to COVID. Then, looking to here, especially, there should be a balance between SARS-CoV-2 and neutralizing antibodies in the system. But as I mentioned previously, we have very less number of neutralizing antibodies even after 5 days or 7 days. Okay, so it's going to infect very badly the upper respiratory tracts. So especially if you have infected with 1 or 3 days, and if you're going and doing any very extreme physical exercise, so you are going to inhale the virus very deeply inside the upper respiratory tract into the lower respiratory tract. So the decline of salivary antibodies is a thing, and this deeper breathing facilitates the inhalation of the virus, and then gets inside, and then pneumonia. Then after pneumonia, look at here, exposure. Okay, then immunoglobulin M is the first antibody is coming into the site of action. Then A, then G, then complications, then incubation, then exit. If the immune system is good, it'll exit. Otherwise, all other organs like myocarditis, like heart, pulmonary thrombosis, nephropathy, all are getting infected. Okay, so if you look at here, massive viral load can overcome the normal natural antibodies and penetrate the lower airways. Okay, so it should not go into the lower airways. If it go into lower airways, a lot of risk is there. Okay, so after COVID-1, we have learned so many immunology terminologies, immunity passports. So initially we had passports to go to other countries. Now they will ask you, do you have immunity passport? Okay, just like we are also hearing immune system overdrive, cover drive, overdrive. Okay, overdraft in the banks. Okay, so cytokine storm, protective responses, overreactive responses. Look at here, immune responses goes into overdrive. All these air sacs where oxygen transfers from the lungs into the bloodstream. So T cells and B cells, look at here, the neutrophils, all are getting dumped inside this, and finally, after cytokine storm, it enters into the bloodstream, and transfer of oxygen can continue with minimal disruption as immune cells. And hyaline membranes, endothelial membranes of these proteins and dead cells lining the alveoli, making the breathing difficult or very much impossible. These cytokine molecules stimulate the movement of cells towards sites of inflammation and infection and trauma. So this is what is going to trigger, and still this is not fully understood. Some scientists have suggested that hyperactive responses from the patient's own defense mechanism can cause a fatal damage. So this is the lungs, which I'm showing here. Is the MERS and other viruses, when it gets inside, at chemokines, macrophages, and so many things. And these are the chemokines, interleukins, and tumor necrosis factors. Okay, and they're going to create so much of mess, and ultimately, apoptosis is the final thing. ARDS, apoptosis, final deadline. The cell has to die uh, with the help of tissue disruption and oxidative stress and SOS. Okay, so free radicals are also generated inside the lungs. So virulence factors, so many virulence factors are also along with the viruses. These bacteria, once along, they come along with the viruses, they alter so many signaling transduction pathways inside the cell, and so many systems are getting hijacked, signaling systems, and then finally disruption of epithelial integrity in the lungs, because lungs are targeted by both bacteria and viruses together in its own strategies, in different ways of strategies. Okay, so just like I mentioned about viral clearance and lung epitheliums, and look at here the clearance. Okay, especially the viral part. Okay, so it takes up the JK1 and JK3 pathways. And cytokine storm is how the host factors. There's so many host factors involved. Okay, so there are two kinds of receptors, ACE1 and ACE2. I'll talk about that. Okay, but ACE2 is very important. And if you talk about the host factors, age, smoking, diabetes, glucocorticoids. If you're already using glucocorticoids, especially diabetic people, these molecules goes and cross reacts with the viral load, and then makes a havoc. So pathways leading to, just like I told you, cis signaling and trans signaling, everything is getting hampered. And finally, the coronavirus is taken up, and whether macrophages, dendritic cells, monocytes. And finally, okay, with the cis and trans signaling, cytokine release syndrome is developed at the end of the 14 to 21 days period. Okay, so if the virus enters inside, okay, so with the help of S1 and S2 proteins, endosomal pathways, the clathrin endocytosis takes inside, okay, and then your unfolding and uncoating, and then early transcription and late transcription, early translation, late translation, and then all your proteins are produced, all the viral proteins are produced, the host cell is destroyed, and finally budding and everything takes place, and exocytosis. Finally, it exits out of the system. Okay, so look at here, the attachment receptors. Okay, so here attachment is SARS-CoV and COVID-2. Okay, so receptors are same for both of them, and these receptors are also same for both of them. I'll talk about this receptor also. Okay, And here, neutralizing efficiency is very high in COVID-1, but neutralizing efficiency is very less. In terms of neutralizing antibodies cannot fight against COVID-2. This is a major drawback with COVID-2. So look at here. So ACE2 is the receptor. Okay. So ACE2 receptor is sometimes called as a devil, or okay, is a boon or a bane. Okay. So sometimes ACE2 receptor, the major function of ACE2 receptor in the human system is to maintain the blood pressure, okay, so the blood circulation and maintain the blood pressure, okay, and it acts, it's nothing but angiotensin converting enzyme, okay. So this has a basic cellular function in the human system. But what exactly happening is, unfortunately, virus has a COVID-2 has got a binding site on the ACE2 receptor. Okay, this attaches and this activates. This receptor, okay, is helper receptor. So if any of this receptor is not there, okay, virus cannot get inside. So both receptors should be there. One attaches, one activates. Okay. So what this virus is doing, virus is binding to the ACE2 receptor, which is very essential for a human vital functions. Okay, taking along the ACE2 receptor inside the cell, demolishing the ACE2 receptors, okay, and depriving or depleting all the number of ACE2 receptors in the cell and killing all the receptors and making the cell unavailable for ACE2 receptors or any other vital cellular functions. Okay, so here what happens is the spike proteins, ACE2 receptors and TMPRS2, okay, and these receptors gets inside, release of mRNA, transcription and replication, linked to stranded RNA, translation occurs in viral protein synthesized. This positive stranded RNA is going to destroy the whole lung epithelial system. Okay, so this is how it gets outside and okay, exists. So when we talk about ACE1 and ANGL2, angiotensin, okay, 81R versus, we look into the receptors of ACE1 and ACE2 receptors, okay, there should be an equilibrium or a balance between both of them. If there is no balance between both the receptors, actually ACE2 receptor is very crucial, okay, and if that receptor, if there is imbalance between ACE2 and ACE1, because virus is going and picking up the binding the ACE2 receptor and taking away inside the cell, and there is an imbalance occurring between ACE1 and ACE2. Okay, so what exactly is happening is all these system is getting infected. Look, like calcium pathway, okay, NADPH levels are getting depleted. NF kappa pathway is completely getting blocked. Then nitric oxide mechanism, okay, which is very helpful for vasodilation. Okay, and SHP2, then FOXO receptors, all these pathways are getting blocked. Okay, and especially vasodilation, anti-inflammatory, anti-fibrotic, and anti-apoptotic. All this system, there is going to get a kind of an imbalance between both the receptors. Okay, so look at here. Okay, so this I already mentioned about SARS-CoV-2, how the host ribosome and release of nCOVID gene and replication, transcription, complex system. Okay, there's so many capsid proteins which are coding. This genome is coding for nucleocapsid N and spike S1 and S2, membrane and envelope proteins, and these are getting binded and post-translated in the endoplasmic reticulum, and then finally going to Golgi, and the Golgi is going to wrap it around with the human cell membrane and going to exit it outside. Okay, so the same is the case, the lines of attack. Okay, so where and how it's going and attacking. The entry site where monoclonal antibodies can be, or convalescent plasma can be used, or chemostat mesylate can be used to block the entry or attachment. And here site 2, where the chloroquine and hydroxychloroquine helps especially. Okay, even here lopinavir also helps in okay, proteolysis. We can block the proteolysis, and remdesivir, we can block the replication transcription complex RNA dependent RNA polymerases. Then translation and RNA replication can be okay, blocked here. And finally, look at here, this endocytosis forming a wrapping around the system. Only that function can be blocked by chloroquine and hydroxychloroquine. Okay. So how long does a vaccine make to take? And people are saying that there are nearly around 20 vaccine candidates already in the market, not in the market, in the clinical trials. We do not know exactly because the whole world is looking at only these receptors, because only these vaccines are prepared. All the 20 vaccines, peptide-based vaccines, DNA-based vaccines, recombinant DNA vaccines, subunit vaccines, nanoparticle-based vaccines. So many vaccines
Sir, can you hear us?
Sir, can you hear me? Hello.
Yes, sir. Yes, sir.
Yes,
Uh,
sir, we are
uh,
hearing you,
uh,
sir.
yeah, shall I go ahead, sir?
Sir, you can continue, sir.
Yeah, fine, fine. Thank you very much. Thank you very much. So it's nothing but shooting at a moving target. So what exactly is happening is probably if you cross phase one, phase two, phase three clinical trials, and by the time the vaccine is coming out, and that vaccine candidate may even have a chance to recombine or mutate with the already circulating strains in the environment, and that serotype cannot be even picked up by anything else, because that's going to become a major problem, because people are all praying that vaccine should work, should not, the vaccine candidate should not mutate, because all the viruses which are prepared, all the vaccines are prepared, are mostly live attenuated vaccines, or not kill vaccines, live attenuated vaccines. So that live attenuated vaccine will have the antigenicity still. If any of such property uh, would mutate or recombine with the already circulating strains, that becomes a major problem. So that's the reason why people wants to withhold with the fingers at crossed, okay, that it should not mutate at ACE2 receptor specifically. Okay. So if you look into the vaccine adjuvants for a strong boosting system, the adjuvant in the vaccine system, look at here. There are toll-like receptors. We have got very less neutralizing antibodies. Gets inside. It's called a depot effect, or a APC, antigen-presenting cell recruitment. Okay, with the help of alum cells, adjuvants. Okay, gets inside with the help of an inflammasome activation complex. Okay, um, after getting inside, okay, antigen-presenting cells will go and present, okay, to the major histocompatibility complex with the help of IL-12, IL-4, then T cell receptor, then naive T cells. Look at here. If TH1 and 2H, there are two lineages. If the lineage is taking TH1 cell, that means it's opsonizing antibodies. Okay, so we want all the vaccines are prepared in the form of opsonizing antibodies, but we want the response in the form of neutralizing antibodies with a strong memory or immune response. Okay, we don't want something to be something like a bubble memory to remain there for a while and then get away. Okay, we are all looking out for a very strong plasma or immune memory responses. Okay, so that it can stay at least for 2 or 3 years. All the vaccine candidates should stay in the body for 3 years in the form of neutralizing antibody, but all the vaccine candidates are prepared in the form of opsonizing antibodies. Okay, so let me not go in detail because it's a hardcore immunology, so it's completely vaccine immunology. So let me not go in details. Okay, so I'm talking about where exactly the virus can be blocked, where it can be, how many vaccines like viral vector vaccines, DNA vaccines, and RNA vaccine, live attenuated vaccines, protein-based vaccines, immune responses, how it's going to be there. And if you look at the stages of vaccine development, there are so many stages: phase one, phase two, phase three clinical trials, then humans, then ACE2 receptor, angiotensin converting enzyme. So many things are there. Okay, so as of now, there are 20 vaccines so far, almost in several trials, phase one, two, three. There are several trials. Okay, we have seen so many news coming up from Oxford, from Israel. People say that we have an antibody, we have a monoclonal antibody, we have one convalescent plasma therapy. They say several things, but I'm pretty sure virus is much more intelligent than all the people living across the globe, because virus has already learned the art of living inside the system and getting mutated every second with its own kind of a differential mechanism. Okay, so this is how it is, and so many interferon-based antiviral agents also preparing, but I'm pretty sure it's much more intelligent than our immune system, and then the scientists, thousands of scientists who are working across the globe in various countries, working day and night to prepare the vaccines. So many companies, drug targets, or companies that target one and stage one. So many things are there, but I'm pretty sure it's only for sake of doing research. Okay, I'm pretty sure if after doing all these, after getting 100 molecules also, if the virus just turns back and mutates for a while, okay, I'm pretty sure they're going to lose the battle, and so many people are going to die. Okay, Okay, so so even convalescent plasma therapy is also suggested in Delhi and Kerala, but not in other states yet. So thank you for your attention. If you have any questions, I can take because I have a meeting with Vice Chancellor. I cannot spare much time in the morning because I'm already in several administrative positions. Thank you very much for your patient listening, and thank you, Ashok and organizing team, for giving me the opportunity in front of you and share my knowledge, whatever I knew. Thank you very much.
Thank you, sir, for your enlightened talk on the host
Yeah.
immune response of COVID-19.
Yeah.
Now the session is open for questions. The people who are in the Zoom link can directly ask the questions to the guest speaker, and the questions from the YouTube live chat box will be presented by me to the guest speaker. Anyone ready to give the
Ashok, you should spare me. You should spare me because I have a meeting with Vice Chancellor because
so
this
you are
time is
you
very precious in
I
the
think you
morning.
are
Yeah.
busy with your daily routine.
Yeah, because
Uh,
yeah,
just for
I'll
to
take the questions. I'll definitely answer all the questions in the YouTube link provided
okay.
to me. Probably during the late evening hours. Yeah, and I look forward to deliver some more lectures to your college because I don't know because most of the times what happens is Ashok. Most of the times the target audience are either arts departments, and for sake of photographs, for sake of documentation, they make them sit forcibly, and without knowing that the target audience are from the same background, and I keep on speaking to them at the code level, but ultimately I'll end up speaking to the walls.
Okay.
So this is what is happening because the knowledge is knowledge. Definitely, I'm pretty sure I've delivered the goods to the gathering sitting out there.
Okay, sir. Here few questions from YouTube chat box. I will mention the designations and the institutions too.
Yeah,
Okay, sir. Here is the question from YouTube chat box.
yeah.
Uh, he's Dr. Praveen, Associate Professor from CHIPS College of Pharmacy, Guntur. He would like to ask, uh, what are the approval technologies related to nanotechnology for the treatment of COVID-19?
I think nano is basically only for delivery system. So if you
Okay.
have to, if you have a vaccine epitope in the form of a protein or in the form of DNA, in the form of a subunit, okay, so that can be tagged with the nanoparticles, and then only that can be used only in the delivery system. Probably you can package all of them in a liposomes, and then only for delivering you need a nanogun or a nanoparticle, or because painless vaccines, or only for the dispersion or sustainable release. Only for that reason they use nano-based formulations, nano-based vaccines in terms of, because the actual candidate is your protein or DNA or RNA, not anything else. Not DNA, it's RNA. I hope
Okay, thank you,
I
sir.
answered.
Here is one more question from Dr. Subir Kumar, Assistant Associate Professor, ASN College of Pharmacy, Tenali. Regarding the currently available diagnostic kits, can you explain any principle related to the biotechnology and which is more feasible as well as a reliable one?
Yes, as far as my knowledge goes, it was very less time because if you want to prepare the antibodies, okay, you have to take the antibodies from a healthy individual, purify them, okay, and purify them, and because I don't think that antibodies from animals can be used against testing the humans in terms of, because I'm pretty sure any neutralizing antibody, if you have to pick from human beings, I'm pretty sure you have to take their plasma, take their blood, and then prepare and purify them with the help of affinity chromatography. I don't think, did the world really produce that many kits in a very short span of a time? And did the people really give that much of blood, donate the blood to the companies? I don't think so because I'm not really happy about, convinced about the kind of results what we get every day. Uh, if at all, if it has to be done, only molecular diagnostics, only real-time PCR is the only best method. And unfortunately, we have very less people trained and people in India who are well versed with virology, because working with biosafety level 3 cabinets and working with very infectious viral nucleic acids, we don't find people in India.
So before closing this question session, Once again, I am requesting anyone from the Zoom can directly pose the question to the guest speaker. Okay, then, as we have no more response right now, anyhow, with the permission of the guest speaker, his email address will be displayed in the YouTube link. Those who have having the specific questions can directly mail to the sir to get your respective answers. On behalf of Vishnu College of Pharmacy, the management, staff, and all, and with the blessings of all the assistant institutes as well as the peer institutes in and around us, we made this session a very grand one today, and I am going to declare today's session has been closed, is going to be closed. Thank you to one and all for your participation, and especially a humble thanks to the guest speaker, Veer Brahmachari Garu, for enlightening us more on the this COVID-19 epidemic and making this session very technical with his delightful presentation. Thank you, sir, once again. And we are more delighted to have you have in contact with you further for our future things also. Thank you once again, and thank you all the participants once again.
I'm signing off. Alvida.
Thank
Thank you.
you. Thank you.